Welcome back to my course Aspects of Biochemical Engineering. In the last lecture, I tried to discuss the broad classification of the microorganism, how the microorganism can be classified, and what are the different microorganism is available. And this lecture, I am going to talk about the how we do the nomenclature of the microorganism, how we name the microorganism. Then also we will discuss the how this organism that grow in the different media, and what are the different components that are required in the media for the growth of the organism, how different different chemicals that influence the growth of the particular organism. And then also I am going to discuss that how this organism can produce the different type of products. So, if you look at the first, let me talk about the taxonomy of the microorganism. This is very important because how we name the microorganisms. This is the science dealing with, because science dealing with description, identification, naming, and classification of the organism. So taxonomy deals with like this, that that when you put the name, then this we take into account the description, identification, naming, and classification of the organism. The Linnaeus development of two words, that is in Latin. One is called genus, another we are called species. Genus capitalize a group of similar organism with different characteristics. Here I want to point out, when we write genus, always we start with the capital letter, but when we write the species, we start with the small letter. This is the basic difference we have between the genus and the species is the smallest and most precise classification. So this is the basic difference between genus and the species. Now I can give the example of Escherichia coli, largely used in the biochemical process. Now Escherichia is the genus name and coli is the species name. So you can see this is Escherichia. It is we have this is genus, and E. coli is the species. Now if you look at that, that classification of the microorganism, in the last lecture also I tried to talk about the classification. That is different type of classification on the basis of prokaryotics and eukaryotics. Now here the classification is on the basis of other types. The one is called phenotype, another genotype, and analytical classification. So these are the now we are slowly go little bit deeper into this microorganisms. Now if you look at this phenotype, what do you mean by phenotypes? Phenotypic classification is based on morphology, physiology, physiological, biochemical, and serological feature. So that we see the organism under the microscope, how it looks. Then the physiological characteristics we take into account. Then biochemical, we can do some kind of biochemical analysis through which we can find out what is the characteristic. And serological means we take out the liquid and analyze and find out what are the different specialty we have. So on the basis of phenotype, characteristics can be done. Now previously here I want to point out that early days, that identification of the microorganism mostly done on the basis of phenotypic thing. Now nowadays we usually do the classification on the genotype classification. We want to have more definite that what organism it is. Definitely we want to tell that what is the particular organism is present there. So it is based on the relationship upon the common ancestry, because what is the heredity? What is the heredity of this? On the basis of heredity, we try to find out do the classification of the organism. Then analytical classification is based upon the detection of structural component and metabolic product using the chemical methods. So that now the microorganism can be classified in three different ways. One is phenotype, also genotype, and also analytical. On the basis of that, we can do the classification of the microorganism. Now, here I want to elaborate little bit. Now, the microorganism can be classified based on the mode of nutrition, growth condition and oxygen requirement, etc. Now, first scientific hierarchy for classification includes this domain, then kingdom, phylum, class, order, family, genus and species. As for example, we have given the example of Escherichia coli. Now, if you look at the Escherichia coli, the domain is the bacteria. The bacteria is the domain. Bacteria is the domain. Then kingdom non-assigned to bacteria, and then phylum is the proteobacteria. The class is the gamma proteobacteria. Order is the enterobacterials. Then family is the Enterobacteriaceae. Then genus is Escherichia and species is coli. So, this is how the complete classification of the microorganism can be given. Now, this is as I pointed out, the classification also can be made on the basis of the nutritional requirement or in the process conditions. As for example, nutrition, then temperature, then pH, then some other factors also we can consider. First, let us consider the nutrition. Nutrition is as for example, photolithotropic organism. This is the autotrophs. Autotrophic organism means they use the carbon dioxide as a carbon source. This carbon source is the carbon dioxide. So, they fix the carbon dioxide. Now, here I want to stress one point that I told you in the last class, that we have several environmental concerns nowadays due to the availability of more carbon dioxide in the atmosphere. Now, due to the presence of carbon dioxide, it causes some kind of greenhouse effect that increases the temperature of the atmosphere. This is the great concern throughout the world. So, this organism has the capability for fixing this carbon dioxide and form the biomass. So, they can remove the carbon dioxide content from the atmosphere. So, they use the light energy, and the examples are algae, purple green sulfur bacteria and cyanobacteria. Then photoorganotropic heterotropy, this is light energy. They use the organic carbon source, purple non-sulfur bacteria and green non-sulfur bacteria. We have green chemolithotropic organism. We have autotropic chemicals that is used as the inorganic chemicals used mostly as the energy source, and then they fix the carbon dioxide. Examples of sulfur oxidizing bacteria, nitrifying bacteria and iron oxidizing bacteria. Now, if you look at the chemoorganotropic, the heterotrophs, this is they use the chemical energy, the organic compound as a source of energy. Then organic carbon they use as a carbon source. Example is the protozoa, fungi and most non-photosynthetic bacteria. So, on the basis of nutrition also, you see we can do the classification of the different organism between the algae, then you have bacteria, then fungi, that all that can be classified. Now, let us come to the temperature. How on the basis of temperature we can do the classification. We have different type of bacteria. We have psychophilic, psychophiles, we have mesophiles, we have thermophiles, and we have hyperthermophiles. Now, very interesting, psychophiles is usually they grow at very low temperature, that is particularly in the cooler region. And I want to tell you that particularly in the Himalayan region, our ambient temperature is quite low. So, now there is a great concern that how we can disintegrate the waste material, because most of the organism that degrade the organic material, they are mostly mesophilic or thermophilic, but not psychophilic. So, a lot of attention has been paid on the psychophilic organism, how this organism can grow at low temperature, so they can safeguard our environment. The mesophile, we have lot of examples. We have bacteria, fungi and cyanobacteria. And thermophiles, we have bacteria, fungi, cyanobacteria also found in the thermophiles. They work at the very high temperature, 45 to 60 degrees centigrade, and then 40. You see, this is 40 to 80 degrees centigrade that we have, and hyperthermophile is 70 to 105 degrees centigrade. This often include the archaea and few bacteria. This is extremophile, particularly in the volcano, that we can identify the organism which can be of hyperthermophile, because in the volcano we have very high temperature, and organisms that survive there that withstand very high temperature. Now, on the basis of pH also we can classify the organisms. We have the examples of this extremo acidophiles. Extremo acidophiles, they grow in between 0 to 2. You can see this is the 0 to 2 that we have. The acidophiles usually grow in between 3 to 5. This is the acidophiles that we have. Neutrophiles usually 6 to 8. This is 6 to 7 or 7.5. Alkaliphiles, this is usually 8 to 11 that we have. Extreme alkalophiles is usually 11 to 14. This is so on the basis of that, that pH also we can classify it. Now, extremophiles, we have the examples is the archaea and the bacteria. Then acidophiles, we have archaea, bacteria, and eukarya. This is I can give the example that wastewater treatment processes, particularly anaerobic digestion process, we use the archaea for the product for conversion of organic waste to methane and carbon dioxide. That is used. The neutrophiles is the archaea, bacteria, and eukarya, that is have alkaliphiles. This is the archaea and few bacteria, and also extremo alkaliphiles is the archaea and few bacteria. This is available. Now, on the basis of oxygen requirement also we can do the classification. We have different type of organisms. We have obligate aerobes, obligate anaerobes. But what is the difference between the what is obligate? Obligate means they required molecular oxygen for the growth and metabolism of the organism. And because if you say obligate aerobes means they required molecular oxygen, not the oxygen in other form. But if you look at the obligate anaerobe, they cannot withstand the molecular oxygen, because if little molecular oxygen present in their system, it will rupture the cell. The cell will be killed. Now, question how it happens? Because we know that it doesn't have the enzymes like catalase or superoxide mutase. Because catalase, we know that when oxygen taken by the living cell, it converted to hydrogen peroxide, and hydrogen peroxide is a strong oxidizing agent, and this is usually degraded by catalase enzyme. So, but this is lacking in the obligate anaerobes. So, and not only they don't have the enzyme like superoxide mutase, because some superoxide formation is there. Therefore, the degradation of the superoxide, that the enzymes are missing. So, if any the highly oxidizable matter present in the system, there is every possibility that biomolecules present in the living organism, their characteristics will be changed and they will be killed. So, and there is another organism, what you call facultative anaerobe. Facultative anaerobe is a typical organism which can grow both in presence of oxygen or in absence of oxygen. Because here you can see most of the bacteria and certain fungi, they have this unique character. I can give you one example, that if you look, I was talking about the Saccharomyces cerevisiae. Saccharomyces cerevisiae under aerobic condition, it produces the Baker's yeast. Under anaerobic condition, it produces the ethanol. So, so it has the dual characteristics. Now, we are working with the hydrogen production process last 2 decades, and we find that Klebsiella pneumonia is a very potential strain for hydrogen production, and this organism has similar type of characteristics. Under aerobic condition, it produces the cell mass, but under anaerobic condition, it produces the hydrogen. Now, then we have aerotolerant anaerobes. So, this is the aerotolerant anaerobes. So, we have they are indifferent to the presence of the oxygen. that is examples of few bacteria and the yeast cells. And micro aerophiles, they require minimum level of oxygen for their growth. They don't require huge amount of oxygen. Micro aerophilics, they are require very small amount of oxygen for the growth and metabolism. Now, that another interesting thing, that how this organism plays the important role, because this organism can lead to different different application in our day-to-day life. As for example, some organism can be used for the production of food products and the beverage. As for example, I can give the example of this, that in the last lecture, I tried to point out that cheese making industry. The cheese is a kind of product through which you can preserve the milk, protein and fat for longer period of time. So, in beverage industry, I talk about the alcoholic beverage like beer and wine that is produced through the alcoholic fermentation process. This is considered as a non-distilled liquor. Then hazardous waste management, that is largely used. I told you that whatever after any kind of most of the biochemical and chemical industry, they pose the environmental pollution problem, because they if you throw the this waste water to the water sources, then what will happen, the whatever bacteria, whatever microorganism present in the atmosphere, they will grow in the water and they pollute our water stream. So, to safeguard that, we shall have to treat the water, and so this microorganism can be, I mentioned in the last class that more than 70% wastewater treatment processes are controlled by the biological means. Now, this can be used for the production of vaccine. We have this is lot of application of our day-to-day life, because particularly for the children, we know that polio vaccine, because a lot of children was greatly affected due to polio, and now this has been recovered with the development of polio vaccine. Not only that, we have other several vaccines we produce that to safeguard different diseases. And then therapeutic agents we produce, as for example, I told the examples of penicillin, that is produce streptomycin, different type of antibiotics we can produce from this microorganism. Single cell protein, this is another area, because this microorganism grow very fast as compared to any other living cells, so they can be used as a very good source of protein. Now, question comes, this is not used as a human food. The reason is that most of this microbial protein contains the nucleic acid, and this nucleic acid causes some kind of gout and kidney stone. So, if you want to use this microbial protein for human food, so you have to remove the nucleic acid. If we remove nucleic acid, then we can use as a source of protein. And that is the hormone, that is largely used for the growth of the plant cell, also other different purpose. Then antibiotics, I told you, oxychemicals largely used. We have ethanol, acetic acid, citric acid, different type of chemicals we can produce from this microorganism. So, it has several uses. Then this microorganism provide the inexpensive alternative to the conventional chemical methods. This is very important thing that we have. And further, the byproducts of biosynthesis reaction usually less toxic, non-hazardous than other industrial chemical reactions. Because if you look at the industrial chemical processes, when you get a certain product, with the main product we also produce some kind of byproduct, which is some kind of hazardous effect on on the environment. So, that kind of problem we have very less in case of biochemical industry, and that is why that this process are more or less what we call environmentally friendly. Now, question comes, how you do the culture of this microorganism? How this is the different type of methods we have. I can show you. This is broth culture, this is agar plate, this is plant culture, stack culture, culture collection. So, this is the broth culture. Broth is the kind of media where the organism grow. You can see when the organism grow, it will be little bit hazy, and then the agar plate, we put the agar. The agar is kind of solidified media where we put media there, and when the organism grow, you can find the different colony formation is there. One dot is one colony, and one dot signifies the presence of one living organism. Then with the slant culture, we can do that in the slant, in the test tube. We prepare that. Stap culture we can do, because stap culture usually we do for the growth of anaerobic organism, because inside that we don't have any presence of oxygen, molecular oxygen, so most of the anaerobe can grow it here. And culture collection, this is usually collected in the form of lyophilized cell. Lyophilization is kind of freeze drying technique through which we can preserve the organism for several months or 2-3 years. We can preserve the organism. So this is how the microbial culture can be done. Now question comes culture media. If you look the culture media, microbes require nutrient. I mentioned before that it require nutrients, and the nutrients can be classified in 3 different way. One is called macronutrients, and the micronutrients, and the vitamins. Now macronutrients means which is required in the large amount. I can give the example that in baker's industry, when we produce the baker's, usually we use the carbohydrate, and we use sugar as a source of carbon, which produces the baker's yeast. Now if you look at the stoichiometry, 1 gram of sugar produces 0.5 gram of baker's yeast. So that largely contribute for the baker's yeast formation process. So this is called macronutrients. But micronutrients means which is required very small amount. What are those? These are mostly the metal ion. That if you look our metabolic pathways, we will find different enzymes that participate in the different metabolic reactions, and most of the enzymes, they required the metal ion as a cofactor. So that is called, they required in a very stress amount. That is why we call it the micronutrient. Vitamins also required as a cofactor. This is then these nutrients perform the several function that aids for the growth. Now I can give you the typical example, the micronutrients. We have carbon, oxygen. You see that we have here carbon, oxygen, nitrogen, hydrogen, sulfur, and phosphorus. The carbon required for growth, product, and it provides energy. Because carbon carbon plays 3 different roles. One is for the growth of the organism, I told you. Then it also produces energy. I told you that when we take glucose, it first pass through the endothelin pathway, then to TCA cycle, then it produces ATP, NADH, and this is used as a source of energy. And then it also produces different products. As for example, citric acid, 1 mole of glucose produce 1 mole of citric acid. So but 1 mole of glucose produce 2 moles of ethanol. So if you look at the stoichiometry is very vital. So it produce from the carbon source we get the different products. The oxygen is required for the growth of the organism, because this is the major limiting factors of the aerobic organism, because microorganism can take the oxygen which is dissolved in the water. They cannot take the oxygen which is present in the air like our system. The nitrogen is essential for the protein synthesis, because that is also very much important for the growth of the cell. Hydrogen, sulfur, and phosphorus contribute components of carbohydrate, lipids, and protein, and nucleic acid. Now these are the metal. This is calcium, potassium, magnesium, and iron. The calcium, this is used in cell activation, heat resistance, endospores, and for activity of the number of enzymes. I can give the example of alpha amylase enzyme. In presence of calcium ion, the thermostability of the enzyme that increases. The potassium and magnesium has served the cofactor of many enzymes and stabilize the membrane and ribosomes. Iron, a part of cytochromes, cofactors for enzymes and electron carrying protein. We have manganese, zinc, molybdenum, cobalt, nickel, and copper. A part of the enzyme cofactor help in catalytic reaction and maintenance of the protein structure. So we can here we can find out what is the utility of different component present in the media. Now, if you look at the media, that composition, it can be classified in different ways. One is called simple media, then one is called simple media, then complex media, different synthetic media, selective, differential, minimum, and enrichment media. So, if a simple media means that a general media used to grow or cultivate the normal microorganism of environment and nutrient agar media. Now, complex media, where the composition of the media is unknown, that is called complex media, and the synthetic media means where the composition of the concentration is known. You know the exact composition of the media, that is called synthetic. Selective means the growth only selective microorganism, which is because some organism require the selective nutrient for the growth and metabolism. So, in presence of some specific chemicals it can grow, that is the selective media. Differential media, it is used to distinguish one type of organism from another type of organism growing in the same media, and minimal media, it basically contains some kind of minerals one in which the few or necessary nutrients are supplied, and enrichment media commonly used for harvesting different type of microbes from the same sources. Now, this is how it looks, the pure culture. It looks, you can see the pure culture, it is like this, that the pure culture is similar type of colony you can find out. But when you have mixed culture, that is presence of different type of that we have different type of colonies we have. You can see that that indicate the different type of organisms are present in the culture. So, this is how we can differentiate from and not only that, if you see under the microscope from the morphological study also we can find out that whether your organisms are pure or I can give a typical example of that pure culture that we have. That in case of yeast, suppose we have our desired yeast morphology is like this, and wild strain the organism is like this. So, if you see under the microscope, even if the same yeast is there, but looking at the morphology we can easily find out whether it is contaminated with the other yeast cells or not. From the microorganism we can easily find it out. So, this is now how we do the isolation is very interesting, that by using a micro manipulator we can take, suppose when we do the plating from the one, I told you one particular colony give this is for the growth of one particular cell, you can take it and then you can grow in the media, that that is how you can do that. Then we can stick plate, you can do it. This is the stick plate we do it. We can in the agar media, solid media, we can put the sticking, you can see it, and then spread plate, you can spread it and pouring it, and this is sprayed and this is pouring it, and from that we can grow the culture in this media. And this is the selectively enrichment media. You can see the different colors by using different ingredients. You can have different. We can put the different antibiotics. On the basis of that, we can find out that the growth of the different organisms in the culture media. Now, industry, as per biochemical industry is concerned, that we have two type of strain we use, wild strain and the improved micro industrial strain. Wild strain, that is the organism that is available in the nature, in the soil. We know in the soil all kinds of organisms are available. And industrial strain, we specially prepared for the industrial purpose. This can withstand the adverse circumstances. As for example, if you look at that, it a higher productivity, tolerance of the product inhibition, high genetic stability. This is very important factor, because if you look at that here, this wild strain, if you use the wild strain for industrial purpose, one particular batch will give you good results, another batch will not give the good results. So this is not acceptable by the industry. It should be the high stability. Genetic stability is a very important factor. The acclimatize to utilize the wide variety of substrate that reduce the cost of the product, and also high substrate conversion efficiency. This is helped in the wastewater treatment process, because load of the wastewater treatment processes will be less. Now this is the process through which we can produce the genetically modified strain, or we can produce the industrial strain, not genetically industrial strain. First is the defining characteristics that we have. As for example, we can have the metabolic capability, including the wide spectrum of substrate corresponding metabolic pathway, release of feedback inhibition and repression, the robustness of the culture, that whether it can grow a high, the change of pH, temperature, dissolved oxygen concentration, osmotic load. I can give the different specific example. Suppose when we talk about the alcohol making industry, if the high alcohol concentration we want to produce, that organism might be having some kind of inhibition characteristics or some kind of osmotic shock, because as we increase the glucose concentration, osmotic pressure increases, that may cause some kind of effect on the organism. So those are the things we take into account when we try to develop some kind of this microbial strain. Now let me tell you how we develop the organism. That we start with this agar slant culture. Then this is the agar slant, we produce that. Then slant, this is sec flux culture. This is the flux we do. They put it in the secker. Then we put in the fermenter. This is a small fermenter we have. Then we secondary seed culture, we produce little bit bigger fermenter. Then tertiary seed culture, we will again 50 liter to 700 liter, and 700 liters to 2,000 liters. Then this is we go for the production fermenter. So this is how slowly we develop the culture for the industrial processes. Now this is some industrial uses of some kind of microbes that has been shown here, that I don't like to discuss in details, because I will discussing this different byproducts that there I shall discuss some of this. Only I want to highlight that that Saccharomyces cerevisiae can produce some fermented juice or distilled liquor, is used in the beverage and industrial solvent. Acidobacter aceti can be used for the production of vinegar. Then Phylogeus niger for the producing citric acid. Penicillium chrysogenum can be used for the production of penicillin. So this particular lecture will give you some broad idea of that, how the microbes behave in a different way, how it differs from each other with respect to their nutrient, because the use some organism required carbon dioxide as a carbon source, some organism required inorganic carbon as inorganic material, some organism required as organic carbon as a carbon source. So that how this is. Some organism can grow at low pH, some organism can grow at neutral pH, some organism can grow in alkaline pH. The temperature also, we have psychophilic, mesophilic and thermophilic. So different nutrient also that the organism that nutrient they differ from each other. On the basis of that we can do the classification. So this gives you the broad idea on the microorganism, how the different microorganisms look under microscope, how they grow in the microscope, how we do the culture. We start with slant, then sec flux, then we ferment slowly. Usually the size of the inoculum should be 5 to 10% of the production media. That usually keep in the mind when you go for the development of the inoculum. Thank you very much.